Hello everybody, this is Dr. Christopher White and welcome to this video dealing with the lab exercise Evolutionary Relationships of Vertebrates. So one of the things we need to bear in mind when we are thinking about evolution over time is that geologists only really have fossilized skeletons to deal with. The chances of soft tissues being successfully fossilized are relatively small. So unlike modern studies, which can focus not only on the skeleton, but also on the soft body parts and the genetics, geologists can only really focus on the skeletons. Now, what this allows us to do, because we consistently focus on the skeletons, is it allows us to track certain changes throughout evolutionary history. So remember that we know where the fossils sit on the geologic timescale. So what we can do is we can use the position of the fossil, so we know their relative ages. We can then apply the study of the skeletons of these fossils and then use that to essentially track evolution for the lineage over time. Another thing that needs to be remembered is that when geologists are referring to skeletons, we are referring to both exoskeletons and endoskeletons. So obviously exoskeletons is the skeleton on the exterior. That's going to be animals like bivalves or insects. And endoskeletons are going to be organisms with skeletons on the inside of their body. So we're looking at fish, dinosaurs, and of course humans. So the use of skeletons to monitor evolution throughout the fossil record has primarily focused on tracking similarities. And so the first part of this lab exercise is going to deal with that process and how we use it to essentially link organisms together. So the basic method is that by tracking the number of similarities, you can work out how closely or distantly related two species were. Typically, the more closely related an organism is, the more similarities it will have, and the more distantly related it is, the fewer similarities it will have. Now, there is a slight caveat to that, which is that convergent evolution will result in the same body morphology forming for organisms that come from completely different lineages. So if you remember, the basic idea is for each environment, there's going to be a preferred design. So the classic example is ichthyosaurs, dolphins, and sharks. So ichthyosaurs are reptiles, sharks are fish, dolphins are mammals. Completely different lineages. However, convergent evolution has resulted in very, very similar body designs. And that's simply because it's the best body design for that particular environment. So in some cases, when convergent evolution is taking place, you will see distant lineages start to show more and more similarities because you have these lineages trying to move into the same environment. So that does need to be borne in mind. However, in the vast majority of circumstances, what we'll do is we will track the closeness of organisms to one another based on the number of similarities that they share. So for part one, we are going to be looking at the evolutionary relationships between 5 different organisms. And we are going to be trying to track the similarities between them and thereby use that as a gauge for how closely or distantly related they are. So our 5 organisms are an earthworm, a human, a dimetrodon, a ray-finned fish, and an oyster. Ray-finned fish, by the way, are the type of fish you would commonly think of. So there's going to include fish like salmon or pike, anything like that. So as you can see, we then have a number of similarities which we will be looking for. So do these organisms have an exoskeleton, an endoskeleton? Do they have a segmented spine? So is it split into vertebrae? Do they have the presence of forelimbs? And there is an asterisk there, so I note down at the bottom here, pectoral, so front fins on animals like fish are considered to be forelimbs in this instance. We have, do the forelimbs have a radius and an ulna? And then for the final thing is, what's the total number of yeses? So for instance, when we're looking at our 5 organisms, so exoskeleton. So do any of these organisms have their skeleton on the exterior? So you can look through the list and you can work that out. So it's going to be a no if they don't, or a yes if they do. Next, do the organisms have an endoskeleton? Once again, yes if they do, no if they don't. All right. It is okay for one of these organisms to not have an endoskeleton or an exoskeleton. Segmented spines, once again, if they do have a spinal column of some kind, is it split into separate vertebrae? Presence of forelimbs, do they have them? Once again, I need to note that the front fins of fish would be considered forelimbs in this instance. And then do those forelimbs have a radius and ulna? So you're probably thinking, right, I can work that out for most of these organisms, but what about Dimetrodon? Well, there's actually a link there to a annotated Dimetrodon skeleton, which is going to allow you to check on which bones they do or do not have. Okay. So once you have written yes or no for each of these fields, you will then total up the number of yeses in the bottom row. Now, once you've done that, you will notice that for each organism, there is going to be a slightly different number of yeses. And so obviously the organism that is most closely related to humans should have the greatest number of similarities, and the organism that is most distantly related to humans should have the fewest number of similarities. So for question 2, what you are going to do is you're going to use the answer from question 1 to put the 5 organisms into order, or should I say 4 organisms, because human is already obviously noted at the top here as being the closest species to humans. No shock there, really. So if you can please put the 4 other remaining organisms, earthworm, Dimetrodon, rayfin fish, and oyster, into a correct sequence, then that will help to show you how we use those similarities to gauge the evolutionary relationships between organisms. Okay, so for part B, we are going to look at homologous structures. So it's not uncommon for related species to share similar skeletal structures, and these are termed homologous structures. Now, these structures will share a similar arrangement of bones. However, the size of the bones, the shape of the bones, and also the number of bones can vary depending on the environment in which the animal lived. So, when you think about a human forelimb, obviously we have the humerus, the radius, the ulna, then we have the carpals, metacarpals, and phalanges. Now, we obviously have a set number of those. So, for instance, we have enough phalanges to produce 5 digits on our hand. However, some organisms will have a very similar design. They'll have a humerus, a radius, and ulna, carpals, metacarpals, and phalanges. However, let's say because they're moving through an aquatic environment, maybe having a broader hand is useful for pushing through open water. So they may have more digits, so they may have more phalanges, for instance. And obviously, because they have more phalanges, that means they'll probably need more metacarpals and carpals as well. So even though the organisms do share the same basic design of forelimb, so there's still the humerus, the radius, the ulna, the carpals, metacarpals, and phalanges, there will be some minor differences. Another good example would be animals like whales, for instance. So they have forelimbs and they have a similar design to humans. So once again, there's a humerus, a radius, an ulna, carpals, metacarpals, and phalanges. However, because they're moving through water, the bones of the humerus, the radius, and the ulna are very overdeveloped. So they're very big, but also quite stubby when compared to, the comparative length of human bones. And that's a direct reflection of the environment in which the animal is operating. So don't be too surprised if you see the same bones. However, they are, they'll be arranged in the same order, but there will be some differences. So the classic example of this is an organism like Acanthostega, and this is a Devonian fish-like amphibian intermediate organism. And it has a humerus, radius, ulna, carpals, just like a human forelimb. However, it has 8 digits. Okay, so it obviously has more phalanges and more metacarpals. And this is simply because it gave it an advantage in the environment in which it was operating. Another good example, of course, would be horses. They just have a single digit. So they have fewer phalanges and metacarpals versus a human. So, okay. So for part B, question 1, we are going to be looking at the human skeleton and the Dimetrodon skeleton. So we're going to drop down, and you will see that there is a fully labeled human skeleton provided. Okay, so you can see we have a front view and a side view. Now that's particularly important, especially when you're looking at bones like the sternum. Okay, Notice that although the sternum appears like it is part of the spinal column, when you look at the side view, you will actually see that it is part of the rib cage. So please make sure you know exactly where each bone is located, both from the front and from the side, in order to stop mistakes happening. So for part one, what you're going to do is you're going to look for these bones in the Dimetrodon skeleton over here. And what you should notice is that all of the bones that are listed here are also present on the Dimetrodon skeleton here. Now, you may be thinking to yourself, hold on a minute, this looks like a reptile. Well, they're not. Dimetrodons are part of the Plesiosaur family, which are part of the synapsids, and they are essentially mammal-like reptiles. They are stem mammals. They are right at the beginning of our family tree. However, when you look at their skeletons, you can still see these homologous structures. So you will see, for instance, if we were to look at the forelimb, you can see we have the scapula, the shoulder blade here. Then we have the humerus, the radius and ulna. Then we have the carpals, metacarpals, and phalanges making up the wrist and palm and digits of the forelimb. So even though this is a massively different animal from millions of years ago, we still have significant similarities in our skeletal design. And that strongly indicates that Dimetrodon is a direct relation of ours. Very, very distant, granted, but we are related. So that's going to be the challenge for part one. And once again, all of the bones listed on the human skeleton here will be on the Dimetrodon skeleton. So you shouldn't have any bones missing. If you do, you will lose marks. Another thing I will just point out is that the limbs, both the forelimbs and the hind limbs of Dimetrodon, they came out kind of sideways. So unlike humans, where the limbs are located directly under the torso, Dimetrodons, the limbs came out sideways. And so that means bones like the humerus and the femur might look quite small on this diagram, the Dimetrodon skeleton, but that's purely because you're looking at them end on rather than from the front. So don't be too surprised by that. Okay, so for question 2, it's going to ask you to look at another figure, and in this case, it's going to be this figure here. So this is the organism Erops, and this is a early Permian amphibian, and you're thinking, right, that looks nothing like a frog, and you would be right. But it does have amphibian features, and we know it is part of that family. But it was a carnivorous amphibian, and as you can see, you can tell that, if I just move over to the left here, you can tell that by the fact it has these rows of spiky teeth designed for, latching onto prey and essentially using it to kill them. So, okay, so what are we looking at when we are dealing with the Erops skeleton? Well, we're going to repeat exactly what we did in question 1, only we are going to be looking at the bones of this Erops skeleton, which, as I mentioned, is an amphibian, versus the Dimetrodon here, which is, of course, a stem mammal, or a very early mammal, or should I say, it's a mammal-reptile hybrid. So, once again, look for the same bones on the as listed on this human skeleton, on this skeleton of Erops. The one thing I'm going to note is that the sternum of the Erops skeleton is not present. Okay, it could be they didn't have one, or it could be that that bone is missing from this particular example, but nevertheless, the sternum is not present, so you don't need to label that. All the other bones, however, should be present on this skeleton. And that, once again, is another strong indicator that humans are very distantly related to this group of amphibian-like reptiles. Now, another thing you will notice is that although humans and Erops do have a lot of similarities, and there are a lot of them, even though, morphologically speaking, very, very different organisms, you will notice that Erops actually shares a lot more in terms of general design with Dimetrodon here. And that's a reflection of the fact that Erops and Dimetrodon are more closely related to each other. And we can also gauge that by the fact that they are more closely related with respect to time, where they sit on the geologic timescale. So that's not a big surprise. So that's another situation where By looking at the similarities between organisms, we can say, right, Eops and Dimetrodon are more closely related to each other than Eops and humans. Okay, so on to question 3. So question 3 is asking you to compare the Eops and human skeletons, and then it's asking you whether you believe that the groups to which these two species belong are related to each other in some way. And it's a relatively simple question with a relatively simple answer. It's a yes or no. So pick whichever one you believe is the correct one. So highlight it, circle it, and then we'll move on to question 4. Okay, so let's move down to the correct diagram. Okay, so for question 4, we are going to be looking at this figure, which contains 8 vertebrate forelimbs. So you will notice that one of these forelimbs has already been colored in and labeled for you. So you will see that the scapula, humerus, radius, ulna, carpals, metacarpals, and phalanges for this human forelimb have been labeled, and they've each been assigned their own color. So for question 4, you are going to look at the 7 remaining forelimbs. And for each of these forelimbs, you are going to color in the scapula, humerus, radius, ulna, carpals, metacarpals, and phalanges. All right. In order to do this, you are going to have to assign each of these bones their own color, and you will fill that in the chart here. All right. Now, please use the same color for each of the forelimbs. So as you can see, I have done the scapula in purple. So you would color in the scapula on each of the other 7 limbs in purple as well. Okay. Another quick point, you do not need to use the same colors that I used for the human forelimb. That's just an example. You are allowed to use whatever colors you have available. So even though it may not look like it, each of these forelimbs will share the same bones. So you should be able to find each of these 7 bones in each of these forelimbs. Now there are going to be differences based on the organisms themselves. So the very obvious one is of course D here, which is the forelimb of a horse. So straight away you will notice that it only has 1 digit. So it only has 1 set of phalanges. So obviously that's a very significant difference when you compare it to something like the seal, where there's clearly 5 digits visible. Now, another thing is that in some circumstances, bones like the radius and ulna can actually fuse together to form one stronger bone. And so in the event that you had a forelimb where that had happened, just try and color in half the forelimb in the color for the radius and the other half of the forelimb in the color for the ulna. The other thing is that because these organisms are all related to each other, the bones are going to occur in the same order. So you're not suddenly going to get the humerus occurring before the scapula. The scapula is always going to occur first. One more thing, also please remember that the shape of the bones can also vary. So for instance, if you look here, you can see that the bones for the dolphin forelimb are quite clearly a lot shorter but a lot wider versus the bones for the human forelimb. And obviously, once again, that's a reflection of the environment in which that organism is living. So, all right, so once you've colored in your 7 forelimbs, you are then going to move on to question 5. So for question 5, it's going to ask you to select 3 of the 7 forelimbs. You're not going to select the human forelimb. And for those 3 forelimbs, you are going to describe how they are adapted to the animal's lifestyle. So how does the animal's body mass, the environment it's moving through, how they avoid predators, how has that affected the evolution of their forelimbs? What specific design features have appeared in response to the type of life, the type of environment in which they are living? So in order to keep your answers clear, can you please bullet point them so that I can see which answer is referring to which forelimb? And so when you're looking at this, you're going to have to think about what type of environment is the animal living in. So For organisms like the seal or the dolphin, well, you know they're obviously living in an aquatic environment. So how is that going to affect their bones? So let's think about that situation. Well, in that instance, they're moving through water. Water obviously is far more resistant than air. So in order to move through water quickly, you're going to require really big, powerful muscles. And obviously the bones are going to have to reflect that. So just think about how that would affect the size and the shape of the bones that you end up with. With respect to animals which fly, so obviously we have the bat and the bird, think about how the weight of the bones is going to be affecting how efficient that forelimb is going to be for the purpose required. And then think of an organism like a horse. Well, horses obviously use their ability to move at high speeds as a way of avoiding predators. So one of the ways they can do that is, for instance, by making their legs as long as possible. This obviously means that they can take bigger steps so they can get away from whatever's chasing them more efficiently. So when you just look at the horse leg here, you will notice that some of the bones are extremely elongated versus, for instance, the human forelimb or the dolphin forelimb or the seal forelimb. So you can look at these forelimbs and you can start to see indications of how the environment in which the animal is living has affected the development of each of their forelimbs. And so you're going to analyze 3 of them, and you are going to try and explain how and why these adaptations have appeared in order to make the animal's life easier. Okay, so that's it for this video covering the lab exercise evolutionary relationships of vertebrates. Thank you for watching and have a good day.